before people. But if we want to know if we want to understand how the landscape has changed, we have to think about what was that landscape like before people. And that's what some of us what we practice with paleobotany when we look at fossil plants or plants that people have been associated with archaeology sites. What we can do is we can begin to use that as a tool to understand what were our floras like before people impacted them. And let's just start with kind of an example, and an example that involves something that we all depend on, food. So I put an arrow. Some of you might wonder, well, where's Georgia and where are you from? I'm right here at the red arrow. That's pointed at a place called Milledgeville, Georgia. And if I have a student currently that wants to work on a memoir-like project, I'm going to have them look at the variation and varieties and nut morphology of pecans. These are an important food plant, and they're literally everywhere in Georgia. As you can see, all of ours are not native. They've been introduced. And pecans are like warmer weather. So if you've visited the United States, or you know people who have visited the United States, you know up here where we have the range of pecans, it gets pretty cold. And incidentally, they're very good. You can make cookies, you can make all kinds of food items and baked goods using pecans. So the question is, how did we get these pecans so far north? It doesn't make sense. And so if you go back 10,000 years, okay, we actually had glaciers in North America, big glaciers, very thick. If you could imagine a couple miles of ice, that's how thick these glaciers were. And at this period of time, what we've found is that if you go to sites where people find archaeological sites, where there were groups of particular indigenous people who traveled up and down the Mississippi, we find the shells of pecans. So we know that this distribution that we see of pecans, and I'll go back to it right here, probably is not its natural distribution, but reflects people picking up these pecans, maybe using them as food. Maybe they would travel and use them as food. Maybe they tried to grow them, but eventually they did grow them there. But you can see that people influence this distribution. So knowing if people have influenced the distribution of plants or not is important. The other thing, too, is that some fruits appear to be ones that we think we selectively bred. You know, one type of fruit in the genus Prunus, you're all familiar with Prunus africana. Here in the United States, one of the favorite food is another species of Prunus that we call peach, and it has this great big fleshy fruit. Okay, and incidentally, all of these fruits, all of these genera over here are in the rose family. So the rose family is a family that many people are interested in if you're interested in the evolution of fruit, and if you're looking at systems to understand things like animal dispersal. And each one of these fruit types that has a box around it comes from a fossil site that is present in a fossil site in Washington State that I'll tell you about in a bit. But the other thing you might notice is we have Rubus. So everybody is familiar with Rubus because it's all over the Virungas. We have, you have Prunus as well, and we have Prunus. So in this place, when we go back to the history of some of these fruit types, just by luck, we have the oldest records of the rose family, of these genera in the state of Washington where we work, and I'll tell you about that in just a minute. So it's rather interesting. Africa has the history of humankind in its record. We have the history of the rose family and of these fruits. So the site that you'll hear me refer to during this talk quite a bit is this area we call the Eosine highlands. And the fossil plants that I'll show you are almost 50 million years old. So in Rwanda, you do not have big records of fossils. And so for you to see this, I wanted to make sure that I included a photo that gives you an idea of where these fossils come from. And you can see it's like a stack of papers. If you know somebody who keeps every piece of paper and saves everything, you would know that the top, the most recent thing that they were sent is on top, and then the oldest thing would be on the bottom. And so you have a pile of these rocks that were deposited in a lake with the oldest rocks at the bottom and the youngest ones up at the top, okay? And so these leaves and flowers were deposited in the lake, and they gently floated and then sunk to the bottom and were preserved. So just to kind of give you an idea, when we talk about geologic time, what the scale of this is, and I keep saying scale is so important. Think of a football pitch, football field. Now, you have to understand, you have to educate me, I'm from the US, but as far as I understand, it's either 100 meters or 120 meters. There could be some variation. So I picked 100 meters football field. If that represented 4.55 billion years of Earth history, the final 1.41 meter. So just think roughly maybe just a little less than 1.5 meters of that football field before the present would represent the time of these fossils, okay? So just to kind of give you the scale. And these are what the fossil prunus that we have from Republic, Washington look like. We have the flowers, but we do not have the fruits. And so for many of you, you're familiar with the parts of the flower. You can see here's a nice ovary. There's the style and the stigma. And so these are, again, we have no fruits. Fruits of close to the same age, a little younger, are found in China and also in the Western United States. So here are the fruits, the hard inner fruit walls that are preserved. So the question is, so oftentimes we think about fleshy fruits. Are they animal or human distributed? Did animals influence their evolution to be fleshy and therefore serve as an attractant to carry off the seeds? Or were they human distributed? Fortunately, in deposits of rock, 2.6 million years of age, in China, you find the pits, the fossilized pits of a species of prunus that look very similar to the modern prunus right here. And so colleagues from institutions in China and Pennsylvania State University, Penn State, Pennsylvania State University, measured and statistically compared key characteristics and basically found that the fossil species, which is red, and the modern fleshy peach cluster together. And so what this tells us is that it was not humans, we people did not collect this species of prunus and continue to cross it to produce big fleshy fruits. Actually, it was animals that were involved, that served to select for the big fleshy fruit types that you see in prunus. Okay. The other thing is that these conservation approaches can be transformative to paleontology. Okay. So for instance, I call this slide herbivory. So here you have the insect herbivory showcasing just our silverback in the corner. And we're aware that all of the relationships of organisms in a community hold them together. And you could represent these relationships as lines. And you think about the base of a community being the plants and some of the bacteria that can photosynthesize. We don't know that we're very uncertain about some of the relationships about mammals and the origin of some of these relationships that mammals formed with plants. So just to show you, the vertebrates are in the red. Down here is the base of the food chain. And you notice here would be herbivores. Well, if you notice, there's only just a few cases where we have examples that go back in the fossil record of animals and herbivory. We know a lot about insects, but we know very little about animals. Okay. And we know that animals and herbivory have an incredible impact on communities. And basically, if you're a plant, you do one of two things. You tolerate it. So if you live in a desert, you have physiology, for instance, that allows you to handle those dry conditions. Well, there's also plants that have physiological processes too that are involved with avoiding grazing. The other thing that you can do for tolerance is just you have a lot of meristems, you have a lot of buds. So if somebody eats one branch or eats a few leaves, it doesn't matter. You keep making more with your meristem. We can't address these in the fossil record, but we can with these avoidance, particularly the morphological characteristics. As many of you know, there's biochemical compounds and secondary compounds and plant defenses, and these are the things that many ecologists, particularly those interested in insect herbivory, focus on. For those of us that work with the fossil record, we look at morphological characteristics. And there's no group of plants that provides a more fascinating example of how vertebrates can influence the anatomy and the morphology of plants than stinging nettles. And you're all familiar with stinging nettles. I'm sure that just about every one of you at one point have brushed against or come in contact with a member of the Urticaceae. And so what you're looking at, of course, is the stinging trichomes. And so they have a glandular base and what looks like the needle-like point and it's glassy. So what happens is that if I'm eating this or if I touch this, immediately the needle-like part of the hair breaks off and there's an irritant that's released in my hand or in my mouth if I'm eating this. So if you bite into this plant, you're going to immediately stop. And it's great because if you are a vertebrate, if you are a mammal and you're eating a plant, the plant does not stand a chance unless it can immediately stop you from eating it. There are other strategies, and there's a lot of complex things about the story, but I'm just going to stick to this. And some of you might be aware that, okay, do they also deter insects? Here's an example. This is from Urtica. And you'll notice that an insect has elegantly, very carefully eaten around the stinging trichomes and have just left them, okay? So we know for a fact, based on so many studies that have been done, that insects are not impacted by these stinging trichomes. It appears that they are all linked with mammals, okay? And so here's the case where I talk about the beauty of collaborating, where somebody's experience combined with yours can be used to address the question. This leaf right here is from a fossil, a member of the Urticaceae. You might recognize some of the names of people too on this paper. Everybody was excited about this paper in the United States because it was the first paper we knew of where we had Rwandan collaborators in paleobotany. And we even had a gorilla expert. I mean, how cool is that? But how that all came about was I misidentified this leaf right up here in the corner terribly. And one day I was sitting with Alphonse, and I happened to be going back in some files I had, and I saw this leaf, and I realized that the project he was doing for his memoir, of course with Urticaceae, I realized that this wasn't anything in the rose family. That's how bad I misidentified it. I showed it to Alphonse. I said, Alphonse, what is this? And he goes, oh, that's Urtica. That's Urticaceae. And of course he was right. And it turned out when we went back and looked at some of these plants, we realized that they had stinging hairs. So here's a stinging trichome that's 50 million years old from Republic, Washington. And here is another one. This is Gerardinia, and you can see the trichomes. So when we come together, when we take experiences and we look at modern pieces, places in time, and we look at the past, we can link them together and have a rich understanding that we can apply into addressing conservation issues. Just in curiosity, if you're wondering what those fossil trichomes look like, there you go. So here's one, and here's one. So you can even see part of the glandular base there. And so going back to, if you were to look at the 50 million years ago in Washington State, here are the creatures that would be in that forest. And there's a lot of herbivory going on, but we're not exactly sure of all the relationships. We do know there's things that ate tubers based on the animal fossils, but being able to understand how these animals interacted, how they impacted the communities and created microhabitats, the best place in the world for us to understand that is by working and understanding what our colleagues are doing in the Virungas. And likewise, for them, we can go back into the fossil record, and there's things that we see that help with conservation and understanding what happens when we have warm temperatures to these faunas and floras. And you can see that that interaction between the past and the present and what we need to do in the future all comes together. So does paleobotany have a significant role in conservation paleobiology? It certainly does, we think so. When we look at techniques, and many of you are familiar with Marie Fidel's work. Marie Fidel gave a seminar here at Georgia College the other week on her work. And again, I just, being somebody that's a paleobotanist, I know how to put together plants from little pieces. And Marie Fidel knew how to sit down and think about that and come up with a technique that she could easily access the plant materials that were in gorilla fecal material and identify them. So when we combined our backgrounds and our technologies and the way we look at things and do things together, we are able to answer so many questions. Obviously, as mentioned before, we have questions regarding the conservation of crop plants that require a datum point. We have to understand that. So we always think about these modern analogs. So when I look at the Virungas, to me, I look and I see many things that are going on that in my mind were going on in Washington State 50 million years ago. Probably the most important thing that I'm asked to do is teach and do projects and things that relate to climate change. In fact, last year I taught 4 climate change classes. So I'm always happy to do that. I never say no to that particular request. And it's because plants interact with the environment so completely. I mean, they interact with light, they interact with carbon dioxide, they interact with the water that's in the soil. And many of you have used techniques when you've done ecological projects in your classes that also go back to this point that we can look at the morphology and forms of plants and relate them to climate. All right. There's one particular relationship that's very important, and to me it's one of those things in the natural world that to me it just is it makes my mind explode when I think about it, is that if you take the portion of entire leaves, so here's a leaf margin that's entire, and over here is one that's toothed, you can actually estimate mean annual temperatures based on the number of woody dicots. So it has to be a woody dicot. So the proportion of woody dicot leaves with entire margins, like these, can be used to estimate mean annual temperature. And to me, that's just, it's incredible. That's one of those things that just isn't, and it works. I've had students go out, and I have students this semester doing this. They go out, they collect the leaves, and it works. And so, because in the Western United States, we have all of these rocks that are piled up like an encyclopedia and big volumes of information that tell the history of floras and how they respond with climate, it makes sense that we had people who wanted to develop the techniques of how to measure and analyze variations of leaves and use them as a thermometer, as a proxy for estimating climate. So Leo Hickey here, who was at Yale, was a remarkable man, and he would literally be able to, would go out and he would literally be able to tell you, you could tell him a section of rock you looked at from his dissertation site, and he would tell you, if you saw it, he remembered, even though he saw it 30 years ago. So he knew place and time and form of leaves, and used this to document climate change. And again, it was using this technique that you can see on your screen to the right. So this P is this proportion of entire leaves. So if you multiply that times 30.6 and add 1.141, here's the equation, what you get is the mean annual temperature, and it works. And so Leo Hickey and his colleagues went that approach. So a few very key features were used to estimate mean annual temperature and also mean annual precipitation. Jack Wolf used a complex set of characters. You can actually, on your own, if you would like, go to the CLAMP website, and you can see that there's a computer program and that you use to estimate and map mean annual temperatures and mean annual precipitation, and it's remarkable. But so you're dealing with 31 characters versus a few. So let's just cut to the chase and just say that fossil leaves provide a means to get good estimates of mean annual temperature and mean annual precipitation. And all of you know, and it's something you don't even have to tell somebody, because you're so attuned to looking at plants and seeing how they change in regions of Rwanda. You would figure out that you can get a good estimate of changes in distributions of plants based on temperature and the amount of precipitation. So when you have these records of mean annual temperatures and mean annual precipitation in the past, it allows you to do something pretty special. It allows you to actually model the distributions based on the niche. And of course, the niche is the role, the place that an organism fits in a community. So how this niche modeling worked, and this is something I know some of you have been exposed to, you have the input data. So we have periods of time. You have your present and your past. You put it into the model, and what you do is you're going to produce a map that shows the distribution of the species at one point of time and at a second point of time. And so by taking this, if I want to know what is going to happen to Hygenia if we would, for instance, change and predict where it would go if you changed the temperature, mean annual temperature, another 5 degrees Celsius, you could put that in there, and you take the future and use it to predict the future as well. So it's not just the past, the future. So niche modeling is something that we're just starting to think about in paleobotany. There's been a couple papers. I'm going to show you one of these papers and show you that it appears to really have the ability to be a very powerful tool. Okay. So when as I mentioned earlier, Africa, you have the honor of having the history of mankind on your continent. We have the history of an event in geologic time where it was the highest level of carbon dioxide. I'm going to talk about that. It's called the Paleocene Eosine thermal maximum in just a minute. But this period of time is going to be very important, because this period of time is being used to predict what is going to happen to floras, not just here in the United States, but other places in the world when you increase carbon dioxide and you increase temperature. All right. So over on the right is just General Scott Wing. And Scott Wing had access to being at the Smithsonian. He was out into a region in the western United States, in Wyoming, where you have these large sequences of rocks. So if you haven't figured out, we have an awful lot of flat rocks in the United States, particularly in the West, and we have an awful lot of them that are also in mountains. And so by using these techniques, what he did was he was able to track the temperature through time. And so over here is 55.9 million years ago, over here is 55.5 million years ago. And he literally spent years until he found the period of time where he could document what the plants were like and what across this period of time that we call the Paleocene-Eosine thermal maximum. Okay. The other thing is by knowing the different kinds of plants, where they appear, where they disappear, we look at things like dispersal and extinction. And so, for instance, during this period of time in warming, looking at comparison of floras, NA is North America, EA is Eastern Asia. And so what we have found is that up to this warming period, you had a lot of extinction, but a lot of dispersal. So it appears that, okay, you have extinction, but dispersal is very important. So when you think about conservation and you think about elevated temperatures, you want to think about these providing dispersal places, being able to provide the means for floras and places to disperse. That becomes important. If you look at cooling, you notice that there was a large migration, a lot of dispersal events there associated with cooling. And then finally, if you take to the far left, this is during glacial periods, and you can see there's a lot of dispersal. And so understanding when things evolve, when we have the evolution of new species and speciation with these changes and dispersal is very important. For us, understanding how fast we are approaching the PETM greenhouse world, what are the conservation challenges for plant responses to these new conditions? So what are the plants doing? How are they going to respond? We have to go way back in time to have an idea of what can happen. So we have to go back to the past and look at these fossil floras and the forms of the leaves to understand what could potentially happen to floras today. And does this concept of niche modeling, to be able to use that to use the present and past to predict the future, does that really work? I just want you to understand, I keep talking about a lot, deep time, millions of years, just to give you an idea of the differences between the rate of carbon that's being released now compared to what it was like, millions of years ago, 56 million years ago. Just to give you an idea, think about this rate. In the blue is the Paleocene-Eosine thermal maximum. And so you had 2 billion metric tons of carbon per year being released, all right? And that was fast. And that happened over 20,000 years. Okay, so over 20,000 years, you have 2 billion metric tons of carbon. And that puts us to this incredibly high amounts of carbon. And many people will say, well, in the past, no problem, because look at how much carbon there was 56 million years ago, and we were all fine, we're all here. The problem is the rate. So you can see over here, currently we have 30 billion metric tons of carbon being released into the atmosphere per year. I mean, that is incredible, 30 billion, and you can see that this rate is so quick. How do you respond? So it becomes a question is, when do you have such a high rate that the dispersal of plants and animals, the evolution, all of these things it's too quick, too high of a rate to be able to accommodate. And just to give you an idea of what these, because the amounts of carbon, it's very hard to visualize. I have problems thinking about 30 billion. Okay, so if you take a look again, this is such a key point for any discussions about climate change and discussing adjustment to high levels of carbon. We have 25 to 30 carbon petagrams of carbon a year versus the slow rate of 1.7 petagrams of carbon a year. So if you're wondering what's a petagram or what is this unit, how much is this? Here's an example. So if you go back, there's 25 petagrams today, or 30. Okay. So 1 petagram equals 1 gigaton. 1 gigaton equals 6 million blue whales, or it equals 100 million African elephants. So multiply that by somewhere between 25 and 30, and that is how much carbon dioxide is being put into the atmosphere every year. Okay. So plants are not animals. We shouldn't expect and develop conservation strategies for them. Plants migrate, they evolve differently than animals. And so then what are the conservation challenges for plant responses in these new conditions? So all of you spend so much time, I'm always impressed with the level of conservation knowledge in Rwanda. It's so inspiring to me. And the programs in September, and just how many initiatives and the applications and studies that are coming out of Rwanda are incredible. And one of the things that everybody in conservation is concerned with dispersal, with managing populations, is dispersal corridors and connectivity. And rarely if you look at connectivity and devising these connections to keep the necessary habitats connected for animals or plants, rarely is climate change addressed in those. You know, we don't think about that, but it's rarely is climate change addressed in those studies. And plant and animal migrations are really different. So if we're talking about a plant migrating, it's the pollen, it's the seed that migrates. But when you talk about animals, it's the adult that can migrate. So the adults are sedentary. The adults stay put, and it's the offspring that migrate. The other thing too is what happens when we have more fragmented habitats. And so this is a big question. And so when we look at fragmented populations, things like Hygenia, how do we measure, that? How do we see if we have the ability and the potential, the genetic variation and the potential for those small pockets to be able to respond to any kind of climate change? So how does a plant respond to climate perturbations? So you have the geographic distributions of a species may contract to a refugium available. So there's one small habitat that's just right, that's remaining, that plants can creep into. And that's very important, that there's always one small habitat, what we would call a microhabitat, that's available. Another one is a species may adapt to the new climate reality and it keeps its biogeographic distribution. I think of Rubus. Rubus has more tricks and more adaptations, and it has more evolutionary potential than any plant. And you notice, no problem. If there's a change in climate, they can hybridize, they can reproduce vegetatively. They have a lot of tools that they can use to adjust to new climate. And the other thing is that a plant can shift geographic distribution, moving to another physical area. So it's going to migrate until it finds a place that it's going to fit. And then finally, the species could go extinct. So can we use these niche models as a way to look at the past and predict the future, and do fossils help us? So 50 million years ago, this is what Washington State looked like, and here is Africa. And you can see that Africa has not yet come significantly in contact with Asia or Europe, okay? And and you can see that if you're using niche modeling for the past, you have to understand that the geography was drastically different than what it was today. Okay. So there is a place in East Asia where many of the fossil, mainly the fossil gymnosperms, still persist. So these fossils are ones that we've found. So every fossil you see here is an example from Washington State. Each of these is represented here today in China. Okay. And so here's just an overview. If we would collect all the plants and represent them as a collection, here you can see all of the different plants that we find in the fossil record at Republic. And so this is more of it's a higher elevation, so it's more temperate in its characteristics, as opposed to lower elevation plants, lower elevation floras that have more tropical elements, as you can see here, that immediately stands out are these palms. And so the models actually do predict what we would expect. Okay, so if you would raise the temperatures up to what they were in Republic Washington, when the age of the Republic Washington fossils, what we find is that these plants are moving upwards. They're moving up to higher latitudes. And so these niche models seem to fit with the things that we see with the fossil plants. So the fossil plants and the living plants in the refugium fit. Okay? So how does this apply to conservation questions? I'm thinking in the United States, it's a tale of two parts of the United States. Over here in the eastern part of the United States, it's really tough to conserve things because we really do not have a lot of connected connected areas. The population in the United States is heavily focused in the east, with some exceptions, like for instance Los Angeles, and maybe Seattle and some areas in the west. But you have all of these natural areas. Okay. So with animals, it's a challenge because you have to have those corridors. You have to have those connected habitats. But with plants, you can target these refugia. You don't necessarily have to have a connected corridor. And the other thing is that you have botanic gardens. You have arboretum. You have collections of living plants that every country can establish. Having national botanic gardens to me, is one of the most important things right now, because those gardens are going to be able to maintain and populations and reintroduction programs, and also have seed banks. Okay. So when I think about migration potentials and habits, I think always of these the mountainous areas in Africa. I love this because there was actually somebody has a model of what the natural distribution of Prunus africanus would be. And I find this interesting because, again, I work with Prunus, fossil Prunus and fossil Rosaceae. And it just seems like of all the places in the world that are so key for biogeography, is that people have left out Africa. And the reason I showed that previous slide is, look, here's where they show it coming into Africa, just right up there at the tip. So right there. So in Africa, there's a lot of interesting studies and concepts and conservation issues that are involved with looking at your high elevation floras and so forth. So for 50 million years, we've had these taxa that represent the Eosine in North America, and they show how you can survive in these complex mosaics of habitat. And basically, the models and things that we look at for the way our plants have migrated in North America are really, really applicable. We can identify these regions associated with mountain ranges as really important refugia for taxa, and it's an important conservation strategy to think about in keeping collections and keeping places where you can maintain living collections and living of some of these high elevation plants. And so with that, I want to make sure I left time for questions. Again, this is a really different kind of talk, I think, for your group, because again, you know, we're so focused on now and conservation of populations that we see now, and as I am too. But you know, the ideas of being able to apply what we know from the fossil record and being able to go back and understand what plants do and how they respond to climate changes. Climate is over time. So if you want to answer any questions about distribution changes in time, if you want to see any changes in form and function of plants over time, you have to go back to the fossil record. And in order to understand the fossil record, you have to really, really understand the plants today. And in fact, my doctoral degree was studying living plants. It wasn't even studying fossil plants. So with that, I'm going to stop sharing and take a look at the chat with all of you and answer any questions. And again, thank you for providing me an opportunity to be with you today.
Thank you very much, Melanie. That was interesting. I would like to ask the audience to ask questions. You can feel free to unmute yourself if you have a question. It looks like in chat we have comments.
Okay, let me refine chat.
Yes, so anyone with a question? Maybe I can start with a question myself. I was wondering, you have been showing especially leaves as part of your research. So I'm wondering if, why did you choose to look at leaves as a reference to the fossil records? Is it possible to use like stem or roots?
Oh, Daniel, that's such a good question. And if somebody was listening, they would think that I probably gave you that question to ask. There's so many parts of plants that can be used to address these questions. There's many different parts of plants that can be used to address these questions. To answer the first part of your question, when you say leaves are the most common type of fossil, common part of the plant that is preserved in the fossil record. Very rarely do we ever find an entire plant fossilized. So we find the leaves. And it also turns out it's the leaves that respond to light. The leaves are also connected with the movement of water, transpiration. And so the leaves are often favored are highly favored because they connect with the physiology of the plant. And best of all, they're the most common part of the plant preserved. There's other people who do look at other parts of plants. So for instance, many of you might be aware that there's people who are interested in ice age and tracing glacial histories. And for their records, they can use pollen. And pollen is identifiable, you know, in their material, and is identical to the pollen you see of plants today. So they have that option. Fruits and seeds are important. A friend of mine, a good colleague of mine named Bruce Tiffany, has studied trends in seeds and correlated them with trends of animals, even going back to things like dinosaurs and how they interacted and ate fruits and seeds. So it just turns out that leaves are the perfect tool to use because you always will have plenty of them, and they interact with the environment in a way that those interactions are recorded in the shape and form of the leaf. Okay.
Yes, that answers the question. Thank you.
Thank you, Daniel.
Yes, everyone is welcome to ask a question or give a comment. We have a comment from Laban. Great presentation. That's Laban from Karisoke, I
Yes,
believe.
Laban as in Virginia.
Oh, wow. So, if we don't have questions, that means people have understood the presentation and they like it. So let's take this time to thank the speaker and thank everyone who attended this presentation. We hope we have... Yes, please.
Can I ask a question?
Sure.
Thank you. Thank the speaker for this nice presentation. But I was interested by this, how leaf are used to estimate the heat till for my very first time to hear about that. So I want to know if we can get documents or papers that have been published. If she can share with us. I'm very interested by that. Thank you.
I would be very happy to do that. In fact, my colleague at Arizona State, Kathleen Pig, and I, we did a book chapter on that.
Yeah.
And so we would be happy to give you the book chapter free of charge.
Great. Thank you.
And also other key papers. So let me put down my email address.
Great.
And I understand sometimes it can be hard for you to get things through ResearchGate. Is that correct?
Yeah.
Yeah. So I will be happy to do that. So if you send me an email, I will provide that chapter. I'll provide it to anybody who would like that. And I also will provide other papers. Okay.
Great, thank you. Do we have any other comment or question? All right.
Thank you.
Just a comment from Tassian. Nice presentation. Apparently, your presentation was fantastic.
Well, I'll tell you what, I'm so impressed with the conservation and the work that's being done at your institution, and working with the students in the memoir program is fantastic.
Absolutely.
I mean, yeah. So to me, it's like whenever you do anything with the University of Rwanda, it has to be my very best.
Mm-hmm. Yes, you did a good job. And so let's take it to the final of this talk. So thank you very much once again. And we hope to see you in the next seminars.
Yes, thanks so much. And now that I'm not teaching, I should be able to join you more. So I would be really pleased to be able to join seminars and hear more about the things that are being done. And thank you so much. It really is a pleasure and honor to present it to you today.
Thank you too. Thank you, everyone. Bye-bye. Enjoy your evening.
All right. Thank you. Good afternoon.